This is my demo of Watson and Crick's DNA structure. The left-handedness of DNA is visible by the top left to bottom right direction of the long strands of DNA, or where the phosphate backbone is facing you while you are looking at the DNA. If it is moving from the top left to the bottom right, it is left-handed. If we're moving from the bottom left to the top right, it would be right-handed. Base pairing is the phenomena that occurs within DNA's helical structure. Purines, such as adenine, bond with pyrimidines, such as thymine. They bond through hydrogen bonding. The purines only bind with pyrimidines because of size constraints that contribute to the helical structure. If purines were to bind with purines, then they would be too large and compromise the structure. If pyrimidines were to bind with other pyrimidines, they would be too small to achieve hydrogen bonding, and thus the structure would be impossible. Base stacking occurs where you have multiple base pairs on top of each other throughout the structure. This again contributes to the structure's strength and allows for the hydrophobic center to remain mostly untouched by water. The phosphate backbone is the rigid outer structure you see on DNA. This is caused by the deoxyribose sugar, which is connected to a phosphate group. This phosphate connects to the deoxyribose sugars of other base pairings all the way across the entire DNA structure. This phosphate backbone is what you see when you are looking at the outside of the double helix structure. A ribose carbon is a carbon in ribose that always has one oxygen attached to it. In deoxyribose, as we see in DNA, this oxygen is actually missing from this hydroxyl group, which leads there to only be a hydrogen there. This polarization of the molecule actually increases stability. The major and minor grooves of DNA can be identified easily by looking at the long versus the short sections. However, if you would like to be more specific, you can see that in the major grooves, or on this side of the base pair, the oxygen is pointed up. It's also participating in the hydrogen bonding. In the minor portion of the groove, or the minor groove, the oxygen is pointed downward. It's not participating in the hydrogen bonding. For some examples, we have the following. Here, you can clearly see the left-handedness of this DNA strand. It is moving from the top left to the bottom right, the phosphate backbone that is, as we observe it. Again, in this image, we can see clearly that in the top left to the bottom right, the phosphate backbone travels as we are